All right. Hello, everybody. My name is Mara Scadden. I am the Director of Science for the Marine Resources Council. Thank you for joining us today. We are going to be discussing Florida's mangroves and a new fungal pathogen that has been identified that may be devastating to Florida's mangroves and needs to be addressed. So before I get started, this disease is called mangrove CNP. C is for Cuvularia, N is for Neopestalotiopsis, and P is for Pestalodiopsis. And these are three different fungal groups that we have isolated in these sick mangroves that are working together to cause disease and death. So how did we become aware of this? MRC has the largest state licensed mangrove nursery in Florida, and we have two locations. One, we have a small showcase nursery here in Palm Bay, Florida, at Ted Moorhead Lagoon House that homes about 1,000 plants, and they tend to be our youngest plants, from propagules to 1 to 3 to 5 gallon mangroves. And then we have our second location where we have nearly 8,000 trees, and that is in Sebastian. And so what we're talking about today, this stress event that I'm going to tell you about, it happened in our showcase nursery in Palm Bay. And so in early February of this year, we started noticing that our trees in the nursery in Palm Bay were undergoing some sort of stress. They were dropping their leaves, they were drooping, they were showing yellowing and leaf spot. And so our first assumption was that our plants were under stress and they didn't have enough nutrients to help deal with it. And so we started treating on the basis of low nutrients with foliar feeding, making sure that our trees had enough water and light and that kind of thing. And it wasn't enough, it wasn't helping. And so we reached out to University of Central Florida's Santra lab, specifically Melissa Dinas, to help us with this problem. And so Melissa was able to come to our showcase nursery in Palm Bay and isolate samples from our leaves that showed these three fungal groups, C and P. And so these photos are some of the symptoms that we saw in the nursery. So chlorosis or yellowing, leaf thickening. So you can actually, if you feel the mangrove leaf, it's thicker than normal, drooping, stem lesions, and death. And so we also saw this kind of characteristic curling that you see in this picture on the left with this propagule, which is obviously not normal. And then on the right-hand side, you can kind of see what it looks like in a white mangrove. And so this is from actually our Sebastian nursery. So we do know our Sebastian nursery has this pathogen. However, they need a stress event to trigger. So we like to refer to it as like chickenpox and shingles. If you had chickenpox as a kid, the virus lays dormant in your system. And then as you grow older, if you have an illness or a stress event, the virus can reactivate and become pathogenic and cause shingles. And this is what we're seeing with mangrove CNP. So Melissa's team and the MRC team have sampled mangroves all over the Indian River Lagoon and from various locations in Florida. And almost 87% of these plants have come back positive for mangrove CNP. So we know that it sits kind of dormant in their system. Under normal conditions, these trees can fight off the disease and keep it at bay. But when they experience a stress event like flooding, high temperatures, high winds, a hurricane, they become stressed and the disease becomes active and causes death. And so the MRC nursery unfortunately lost about 80% of our plants that showed signs of stress and mangrove CNP. And so it does have a high mortality. And so this is something to be concerned about because we have noticed it in the wild populations. That with climate change, as tides are getting higher, we're having sea level rise, so our mangroves are more flooded than they're used to. We're experiencing higher temperatures, that kind of thing. These plants are going to be under stress. And so one of the things I wanted to point out here on the left is that in our red mangroves, it's pretty easy to tell if if the tree has been exposed to mangrove CNP. And that's because of this kind of characteristic black spotting you see on the left. And so each of those spots is a plant's stoma or stomata, their gas exchange pores. And when you look at it under a scanning electron microscope, you can actually see that the fungi have burst into the plant and out of the plant. And so we know that the fungi enter the plant through the plant's stomata. And then on the right-hand side is a really, really thick white mangrove. And so this mangrove is down on our shoreline at MRC and has experienced some stress. It's been really flooded recently, but it's been able to keep the CNP at bay for a bit. But the old growth that's been there the longest has become very notably thick, and that's because of the pathogen. But the new growth is normal and thin. And so this is the easiest way that we're able to keep track of this in the wild. And now we do need help because we need to be able to monitor this in the wild. We have reports of dying plants all over the Indian River Lagoon, in Tampa, in Sarasota, up in the Panhandle. We know that this disease is all over. And we also know that it's been noted in other countries. It's been noted in Malaysia, Pakistan, Australia, China, Puerto Rico, the Bahamas. All of these countries and places have experienced die-off events due to mangrove CNP, but there hasn't been a global effort to treat it. And so I'm going to introduce Melissa Dinas to talk about her research into the disease itself and the fungi, as well as the treatment that her lab has come up with called MagSun. So here is Melissa.
Hello, my name is Melissa Maria Denis, and today I'm going to be talking to you guys a little bit about the evaluation of mangrove die-off, what's causing it, and what in the last 2 years we've been trying to produce or create to solve it. So before I begin, I want to give a little background on myself. I began this project in 2018 as a sophomore in high school. And it's kind of evolved throughout that, and that's what I'm going to be sharing with you today. So the significance of red mangroves, I believe I'm kind of talking to the choir, as most of you guys know, but specifically I want to look into what it means for Florida. So red mangroves shelter a lot of our native organisms, a lot of our fish, crab, and bird populations use mangroves as native habitats. Also, they protect our coastal communities. We all know when we think mangroves, we think of stopping floods and really protecting our shorelines against hurricanes and other weathering effects. And lastly, I kind of want to touch upon economic benefit. So when we think of red mangroves and how they protect us in shorelines, we kind of always think about those root systems that are so unique to red mangroves and mangroves in general. But there has been literature and studies shown that that's not the only thing that protects us. Also, the foliage or the leaves of the red mangroves also protects us from negative effects. If you kind of think about it, red mangroves have helped us stop power lines coming down. When winds from hurricanes come in, the roots are not what stops those winds from getting to neighborhoods or places where they can topple things over. It's actually the leaves. So having a high density of leaves on our mangroves is also important, not just the root structure. So since the 1960s, about 50 to 60% of Florida's native ecosystems mangrove ecosystems have completely been wiped out. When I speak upon this, I'm speaking about native. So not anything from the repopulation efforts, just our original mangroves. So it's a very big amount. And when we think of restoration, those mangroves are fairly new, or they haven't aged quite as much as our bigger, stronger mangroves on the coast. So this presentation is going to touch upon plant pathogens. And for those unaware of what plant pathogens are, I'm going to give like a quick little recap. When we think specifically fungal plant pathogens, we always kind of think of this image when you can quickly identify, quickly see that it has these fuzzy mycelium structures. You're like, fungi kind of looks what you get on bread when it goes bad. So this is kind of what people think of when they think a fungal pathogen. But today we will be talking about endophytic pathogens. Endophytic means it lives inside the tree and inside the organism, so it's not as easily recognizable. As you heard before, Mara mentioned her analogy that it's kind of like chicken pox. It lives within the organism, and unless stressed or something occurs to activate it, it'll start showing. So we call these pathogens specifically opportunistic pathogens. Opportunistic in the sense that they wait for the plant to either have other stressors that need their attention and they can overtake the plant, or something happens in the microbiome inside the plant that allows these pathogens to take over. So symptoms of infected leaves in the wild that we've kind of looked at is chlorosis, or yellowing when the chlorophyll of a leaf dies off. We've noticed these leaf spots on the second image, where you can kind of see that it's just little larger black dots all over the leaf, and then necrotic tissue. Another symptom that we've noticed is these little black spots on the back of the leaf. When I was in high school, I had a teacher who also studied mangroves and was like, oh, this is a very easily identifiable feature of a red mangrove, or just mangroves, these black spots on the back. But when I joined the MISA lab, which I am right now, my partner actually said, hey, Melissa, his name is Jorge, why don't we look at this under SEM? I'm so sure that this can't be something that's just happening because we don't see in any other plant. So I was like, okay, let's look at it. And then when we looked at it under SEM, which is just a very high profile microscope, we actually saw that it was damaged stomata. Right now, our hypothesis is this either the entry or exit site of the pathogen. So that's kind of what we have right now on these little black spots on the back, but it is one of the ways we identify right now currently if it has the pathogen. So speaking a little bit on where I've collected, where's all this data coming from, or Melissa, where have you been collecting your samples? I have 3 major location sites as of right now. My first collection site and where this all began was at Fairchild Chadwick Botanic Garden in Miami. We have this mangrove forest inside the garden, which spans about 5 to 10 miles, depending on where you go from. And this is where I first began collecting my leaf samples. We also have, now that I'm at UCF, we have a northern site at Indian River Lagoon, by the Apollo Beach Center. And then we also have what I call our southern site. I chose the southern site not just because it was just more south, but because it was an isolated mangrove island. So this was not connected to any shorelines, and it was for us to see if the pathogen is spreading through coastlines or through shorelines because they can touch one another. It's not that big of a jump to get from tree to tree, or it has infected plants in this island with just water and no other touching mangroves. So how do we know? Me just telling you where I collected my samples doesn't really give you much. But basically, we would go out to these collection sites. We would grab plants and and leaves from these trees, and we would plate them in special agar, special media that will grow whatever's in these leaves. And then from there, we would identify microscopically. So when we identified microscopically, we started seeing trends on what type of fungi are there. But again, if you guys don't know, fungi aren't very easily identifiable by the mycelium, or the like spaghetti structures most people think of. You have to identify them through the spores. And this is actually an image of one of the spores of one of the fungal families we've been finding very repeatedly. As you can see, it's a very distinct structure. I kind of like to call them squids because I think they look like squids. But they have these median cells, which is like the 3 fragments, and they have like an apicable appendage, what I call the tail, and these basal appendages would kind of look like antennas, or like the little appendages of the squid. So again, what we're thinking is that these spores are traveling through water, air, insects. More research has to be found on the vectors of this pathogen, but they're going in through the stomata or coming out to spread the disease. So as of right now, in the wild, we've seen about 57% of Pestiloptiopsis and Neopestiloptiopsis species, which are our major plant pathogens that we believe are affecting mangroves. Our third most found pathogen is actually a Covalaria species. And then this non-pathogenic and unidentifiable slices that you kind of see, non-pathogenic was just fungi that we knew just grows naturally, like penicillium. We had other fungi that are just normal in an environment, and we just quantified those as non-pathogenic. And unidentified was just fungi that either got contaminated with bacteria or just never sporulated, so we were never sure of what it was. So me telling you that we grew all of this for specifically pathogen people is not a sure way of saying that it is our pathogen. I have a low battery. I don't know if you saw that. Okay, let me go back. Cut. So when looking at fungi that we've isolated, in plant pathology terms, that's never a surefire way to say, oh, that's what's causing the pathogen or the pathogenesis in the plant. Well, what we kind of do to confirm this is get healthy plants that we know look very nice, very green, and we actually inoculate the pathogen into them. So as you can see here, and if you kind of remember from the images Mara showed you back in her presentation, we're kind of seeing some trends. This middle picture right here, where you can see the curling or kind of twisting of the leaf, was very prevalent in MRC's instance, as well as the chlorosis, and also this wave-like pattern of necrotic tissue or dying tissue was very prevalent. And then for the Pachylepthiopsis species, once again, we saw this chlorosis, but as you can see, it has like these polka dot structures. That was also very prevalent in MRC's instance. And then most importantly, like this floppy mangrove leaf that we've also seen, kind of like a big gust of wind came and knocked them down, but I promise they were in a little isolated container. So this was just something that we were seeing with the pathogen. So I just told you a lot of bad news and a lot of sad things, what I think are very sad. So let's start talking about what we're doing now to help the problem and what we consider the future of this research. So as I mentioned, I started this research back in 2018, but I started actually here in the University of Central Florida in 2022. So when coming here, I knew I did not only want to find this problem or survey or understand it, but I also wanted a way to mitigate or help it. So I joined specifically the MISA lab, which is run by Dr. Swadesh Sancha, and they specialized in agri-nano formulations or making chemicals for agriculture. And as an environmentalist at heart, I knew I can take these ideas, but twist them a little to come up with an environmentally friendly solution. But happily enough, when I joined, Jorge Pereira, who's actually our senior most PhD student in the lab, he had already come up with what we are using now, this what we like to call MagSun. So a little bit on MagSun, we call our magnesium hydroxide nanoparticle MagSol, and then we tank mix or combine it with sodium polysulfide, which makes MagSun. I will go in a little bit more on how this was not the original synthesis per se. Both of the components were original, but this synthesis was made for bacteria. So we did have to tweak it a little bit to make it more suitable for fungi, as fungi are more complex than bacteria. And we just needed something that can help eliminate the fungi. But so speaking about this nano formulation, we specifically chose magnesium and sulfur because they're both secondary macronutrients to plants. So kind of what the idea behind this was, how about we can take like a vitamin pill that also helps fight off disease. So we're using nutrients that the plant will already use for their life cycle, but tweaking them in a way that can also stop fungal infection or disease spread. Also, sulfur is already highly available in mangrove estuaries, mangrove ecosystems, and they're also a natural antifungal. So this was kind of like the thought process we had with creating this material. So why nanoparticles? Why can't we just use a normal molecular standard pesticide or another material? Well, nanoparticles are necessary to reach endophytic pathogens. Inside the plant, nanoparticles can move through the vascular of the plant and kill something that's within. A lot of molecular pesticides stay on the leaf surface. Also, because again, how I said nanoparticles are so small, they're also a lot more specific or targeted. So we can use lower concentrations and still be able to kill the pathogen because they're so targetable. And lastly, as I said, these are nutrient-based solutions. So it also is more eco-friendly, which when thinking about such a complex environment as mangroves, it's one of our first points that we knew we had to hit. So how do we test this material against the pathogen, or how do we know that it's killing or stopping growth of the pathogen? So I will be talking about two different fungal assays today. The first is a mycelium growth assay. So a mycelium growth assay is that you take a plug of fungi and you put it into broth specifically, it's a liquid media, and then you add your material or what you think will stop the fungi from growing. What is very key to know about the mycelium growth assay is that this shows for inhibiting, not death, but how much you're stopping the growth, not how much like how toxic you are to the fungi. So this is our results. So just so you kind of go with me through this, the control is how the fungi grows naturally. It's no treatment, just fungi in broth. And this 0 hour, or this zero that I have right here, is how I mentioned before that MagSun was originally dedicated for bacteria. So this is what the original synthesis did. And let me tell you, when I first did this experiment, I was like, wow, we're stopping growth by 70%, about 70% around there. We're doing so good. We're amazing. This is the best thing ever. But then looking at it, I'm like, okay, how about if we find a way to stop it even more? And that's when we did this tweaks in the synthesis, and that's how we got treatment 1, 2, and 3. And if you notice, even from the 0 hours, we got a significant reduction in growth. I believe right now it's about 87 to the 90s, around there in growth inhibition. But also, Melissa, they all kind of look the same. Treatment 1, 2, and 3 have about the same milligrams of biomass. And you are correct. So what we kind of saw was at this concentration, which is 512 ppm or parts per million of Magsol, combined with 256 ppm of NAPS, the treatments kind of inhibited the same. They stopped the growth about the same rate. So that's when I come in with my second antifungal assay, which is the poison food. And this is where we start looking at how much we're killing, how toxic we are, which is kind of fun because it's in the name, poison food. So what this kind of is different because you do it on solid agar or solid media. You put your material into the media, you plate it, and you let it dry or become solid. Then you put a fungal plug on it, and the fungi to grow needs to eat the agar or consume the agar to grow out. And if it's toxic, when they eat it, it dies. So that's kind of the difference between these two poison foods. And if we look here, but all three of the treatments did stupendously well. You see here for the one thermal treatment, we had a very big reduction in growth, and also for the third thermal treatment. But when we really see a very significant change is the second thermal treatment. If you kind of look at this last agar plate, we almost got zero growth or a very low percentage, even compared to the other treatments where we also saw a significant reduction. The 12-hour or the second thermal treatment is what did the best. And if you're a numbers person, because some of the people in my life are, you can easily tell here that this orange line all the way at the bottom, which is our second thermal treatment, did the best. It had the lowest growth rate. And then the yellow on top is for your comparison on what no nanoparticle treatment looks like. So how can we apply this to the real world? Honestly, when we began this project, we had this kind of far off futuristic ideas of how we were going to treat mangrove forests in the real world, in real time. But as I said, that's a very far off idea just because of the whole registration process and all the years of data we need to collect to be able to implement this into living shorelines. But what was kind of a happy coincidence was I needed healthy mangroves to run my pathogenicity tests. And MRC, I reached out to them and be like, hey, can I get some mangroves for these tests I want to run? And they're like, yeah, sure. They bring them over to our lab. And I had a very tiny little meltdown because all of their plants were also infected. And that kind of got the ball rolling. MRC was like, what do you mean? We noticed that we've had a die-off, but we really didn't understand why. And I was like, let's look at it, let's think about it. And it was the pathogen. So how about if we not only look at the living shorelines, but at the nursery level? One important fact is that we have preliminary data that shows that this pathogen is in seeds as well. We've grown the pathogens out of seeds. So right now, as our hypothesis, it's that the mother plant can transmit the fungal pathogen through spores to the seed, as it's completely systemic, as I was mentioned. So with thinking about this, we're like, okay, how about if we can sterilize seeds, which then nurseries can use to grow back their populations in a healthy manner? Because there's one thing about a seed already having the pathogen, or it just gets it through winds, which is a very smaller chance. So right here we have seeds that we soaked in the second treatment that I've mentioned, and you saw the data before, for 24 hours. And if you look at the upper picture, you can see that we do have growth. We have a lot of fungal growth. But then just 24 hours after, when I got another section, we see that growth is significantly reduced. And this, I believe, was on day 4 of us viewing the plate. And then we had another plate, which I didn't image, but that one grew penicillium. So we completely lost that very distinct pattern of Pestiliptiopsis on the plate, but we were growing penicillium. So you're not completely damaging the microbiome in the seed, which I know for some are like thinking, oh, how about if we completely die it out? But also, Jorge likes to do this analogy, which is if you take an antibiotic, you're going to kill healthy and pathogenic bacteria, but an organism will always recuperate that microbiome. So that's the idea that we're running with right now. And then we also looked at, okay, how about if we infect larger plants and then we treat it with our treatment? And these plants actually were inoculated at the same time as the other previous pictures I showed you with our pathogenicity testing. So what we do is, again, we inoculate and then we wait 5 weeks to see the pathogenic signs. And these plants, we left them alone. They went through the same process as the other plants that you saw before, but on week 4, we treated them for 24 hours with our MagSun solution. And we saw a great improvement. So that to us is like, okay, maybe this is also a viable plan to treat mangroves in the nursery before they're put out for restoration efforts. And yeah, that's kind of, I know that was a lot, but that was kind of the last 5 years of my life. Saying that makes me feel a little old, but thank you again for listening, and I will take questions if anyone has some. Thank you.